Haemonchosis in sheep and goat, a case study. So as we all know, the Haemonchus concertus, which is mainly causing the haemonchosis condition, is a highly pathogenic parasite that localizes in the abomasum of the affected animal, and its pathogenicity exerts by the blood sucking activity that is adversely affecting the health and productivity of the animals. So it is mainly known as the Barber's pole worm, that is also called as the twisted wire worm or large stomach worm. So they normally feed on the blood from the capillaries that is present in the abomasum of the ruminants. Most pathogenic causing things or the causing characteristics is that the single worm can ingest up to 50 microliter of blood per day. So high infection levels can cause severe blood loss, that is more than 100 ml per day, because it depends upon the number of the worm that is present, and it is so heavily infested that there is millions and millions of the worms that used to be present in a clinical cases. So that blood loss is followed by the anemia and then hypoproteinemia. So this is the photograph I'm showing. It's a representative photograph, that is the twisted wire worm. You can see in the left picture. And the right there you will see the different means lot many of parasites are present in the abomasum. So infected animal have a reduced digestive capacity, which affects the uptake of the nitrogen, organic matter, and the energy, thus leading to the unproductiveness or leading to a very emaciated condition of the animal, and heavy infection may cause animal to death. The newly weaned animals or the small animals are more susceptible, and the other vulnerable factors that increase the susceptibility include the overgrazing, then high stocking rates, and inadequate nutrition leads to more susceptibility of the disease. Then one adult female worm can lay up to 10,000 eggs daily. So if lot many moms are present, means they will go for lot many millions of eggs per day they will shed, and that eggs will be coming out in the feces, and then they will again go to the other animal or infest the grazing pastures. In case of this haemonchosis, there are many challenges that we have to face as a veterinarian. The first challenge is the uneven distribution of the infection globally. That is, the being more prevalent in tropical and subtropical and warm temperature and summer rainfall regions, that is an adequate temperature and then humidity is required for this parasite, than in cold and cold, temperate and arid regions. So there is difference of approach that is required to control for these two warm temperate region and cold arid regions. So that is a first challenge to restrict the infection. Then the climatic conditions that has significant impact on the development of the egg to L3 larvae, that is the infective stage, L3. And humid and warm climate in the optimum conditions for Haemonchus species and infection of the ruminants. The lowest temperature limit is 18 degrees Celsius, and the rainfall is about 50 mm, that has been reported by Gordon 1948. And the highest temperature limit is 28 degrees Celsius, that is over 70% humidity, that has been reported by Rossanigo and Gruner 1995. So, and in case of low temperature below 9 degrees Celsius, there is little or no development of the parasitic eggs. Then coming to the second challenge is the widespread presence of Haemonchus strain resistant to the various anthelmintics available, that is the benzimidazole, imidazoles, macrocyclic, lactone, clozentel, monipetal, which makes the control of the infection difficult. That is mainly concern nowadays. Then third challenge is the difficulty to diagnose the disease. The field evidence, whatever that some symptoms are showing in the field, is not enough to go for diagnosing the disease. We have to go for confirmatory tests in laboratories like fecal test, or beyond that nowadays people used to go for molecular test also. And then this is required to go for diagnosis of a disease. So only in case of field or in case of clinical symptoms, only we can see the anemia, emaciated conditions, etc. or decreased feed intake, these things we can see. But after confirmation with the fecal test, we can go for telling that, okay, this is the disease is going on inside it. Then the final challenge is difficult to control the infection. So a necessity to use the pharmaceutical products cautiously and with a planned approach is required to avoid the further development of anthelmintic resistance. Again, there is a problem of auto-infections also, the reinfections also is there, because the animals are continuously spreading the eggs in the pastures, and while grazing time, they will again reinfest the animals with the eggs. To check the Haemonchosis or to reduce the cause of the Haemonchosis, we have to know the life cycle of the parasite, which is a direct life cycle. 6 stages of life is there: egg, then 4 larval stages, and the adult one. So here in the figure, we can see the adult worms are attached in the abomasum in the goat, and then it is passing in the feces as eggs. So that egg develop in the feces into L3 larvae in about 6 to 7 days in the grass blades. And then that L3 infective larvae move up to grass blades at 2 to 3 inches in the worm, phlegm, or the dew. Means they are going up in the blades so that while the animal is grazing, they will take the eggs. So grazing goat will ingest the L3 larvae, and then that L3 larvae will again go to the abomasum. They will reside there. They'll grow up to L5 stage, and again it will lay the eggs. Then the female parasite lays numerous eggs with an average of 1295 per day, which is passed through feces to the pastures. The eggs may die or develop to free living larval stage, first stage, second stage, infective stage within 1 to 7 days. Then hatchability of eggs and development to infective larvae depends upon the availability of suitable environmental conditions, which I have told, in fecal pellets and herbage. Then L3 stage is ingested by the host, where it undergoes encystment in the rumen and takes 2 to 3 weeks to develop into parasitic stage L4. So after 2 molting and just before the final molt, immature adult L5 erupts, which developed a lancet to penetrate the mucosal vessels for sucking the blood. So as we all know, the abomasum is the predilection site where the adult worms move freely. And the L4 may also undergo arrested inactive phase called the hypobiosis during winter season, or whenever the environmental condition is not adequate for the development of the larvae. So we have to know about this topic, that hypobiosis, this is the unique feature of Haemonchus species, which refers to the inhibited development. The seasonal changes, that particularly the temperature has major role in determining this hypobiosis. The feature is activated when opportunities for transmission of the larvae are restricted, that is cold weather, high temperature, dry season, low humidity conditions, etc. The most suitable condition for the transmission of eggs to larvae in the majority of helminth parasites are provided by warm, wet weather, where the larvae can remain alive for 6 to 8 weeks. So the hot, humid, tropical climate of a country, that is in India, it is suitable for the development and survival of pre-parasitic stages of the nematodes throughout the year. So in this case, in this parasite, the pre-patent period is 19 to 21 days, but it can be shorter also in case of weakly immune animals. So coming to the consequences that has been shown in the animals is the significant reduction in the production of infected animals, then decrease in the growth of the young animals, then decrease in milk production in lactating animals, and in fiber production in case of fiber producing animals, and ultimately leading to the severe economic losses for the farmers. So far, 12 species of this Haemonchus is identified in ruminants, where the Haemonchus contortus is mostly or main pathogenic nematode in case of sheep and goat, along with the Haemonchus placei. This genus Haemonchus species was first discovered in 1803 by Carl Rudolphi and includes the blood-sucking nematode helminths. So the family here is Strongylidae, and that Haemonchus contortus that today we are discussing is the most pathogenic nematode helminth of the small ruminants. Now, coming to the diagnosis of this disease, first thing is the clinical symptoms, which is the anemia. Mostly anemia is showing in the animals, along with the lowered feed intake or the emaciated conditions. And we can see also the bottle jaw condition, which I am showing in the top photograph, that bottle jaw condition is sometimes we are able to see in the clinical cases. Then coming to the fecal examination, we can go for direct fecal smears and then we can observe on the microscope. Then fecal flotation for parasitic eggs, we can do that. Third, we can go for coproculture also to get the L3 stage from the eggs to identify it is actually the egg of the Haemonchus or not. And lastly, we can go for a postmortem examination for this determining the immature and adult worms, that is presence of worms in the abomasum of the animal. So in this slide, we can see the first photograph. It is having the eggs, picture of the eggs, which we'll see in the fecal examination. Then second B picture is the L3 larva with the tapering end, which is the characteristics of the L3 stage, that is the tapering end. Then a third photograph, it is of the adult female, that is having the vulvar flap, you're seeing. And the last one, it is the photograph of the adult male having the copulatory bursa and the spicules. So to identify the worms or to diagnose the Haemonchosis properly, we have to know the microscopy of the worms. That is, the female worms are 18 to 30 mm long and are easily recognized by the Barber's pole appearance, that is the white ovaries and uteri twisting for the length of the worm around a red blood-filled intestine. So in the first photograph itself, I have shown you the twisted wire formation type, that is actually called as the Barber's pole. And whereas the male are 10 to 20 mm long and uniformly reddish brown, the male is totally a reddish one. The body length, cervical, papillae length, number of cuticular ridges, vulvar flap morphology represents the main criteria for identifying and differentiating the female parasite species. It has a tooth or lancet in the poorly developed oral cavity, which helps to perforate the gastric mucosa and suck blood from the capillaries. So coming to the postmortem findings, in case of hyperacute form, we will get the detection of large number of adult Haemonchus species in the abomasum and the macroscopic observation of the petechiae on the gastric mucosa of the organ. In case of acute Haemonchosis, the affected animals appear severely anemic with the pale mucous membrane, ascites, submandibular edema, indication of severe hypoproteinemia, etc. So these bloods, they are sucking that may be unclotted, and gastric mucosa becomes extremely edematous, full of petechiae with visible visibility of the attached parasite. In such cases, the helminth may vary up from 2,000 to 20,000 adult parasites in each animal. Sometimes it used to be much more than that also. So thus the postmortem findings are not indicative of long-standing Haemonchosis. That is, if there is chronic infection of Haemonchosis, in such cases, the small number of adult Haemonchus species in dead animal abomasum, and we used to get in the postmortem examination. So that time we have to couple the history of the reduced productivity and relevant clinical signs, that is the anemia and other conditions at the population level. We can go for that and we can tell it is the chronic stages of Haemonchosis. Now coming to the treatment and control, means treatment is mainly used with the help of the anthelmintics, that is benzimidazole groups having the albendazole, fenbendazole, oxfenbendazole, and then the macrocyclic lactones coming with the ivermectin, levamisole, etc. are being used in the field conditions to treat the animal, which is mostly means very much effective in case of this Haemonchosis. But nowadays, due to the resistance of these anthelmintics due to improper uses, can say overuses where there is not required, also when there is overuses, that it leads to the resistance of these anthelmintics in various places. So that's why nowadays it is restricting the anthelmintic use. Otherwise, the other two methods for control is the non-chemical, and then now vaccination is also coming on the way. So under non-chemical control, it includes grazing management, emphasizing on rotational grazing, the pastures. Then secondly, the modification of animals' diet, which directly affect the response of the animal's immune system to Haemonchosis. Then selection of animals genetically resistant against the Haemonchus species, and administration of various products to animals, that is the nematophagous, fungi, tannins, or polyphenolic compounds. We should mostly emphasize on this non-chemical treatment and control management systems. So here we can give emphasis on the grazing management, that is cell or rotational grazing method. It contributes to minimizing the intake of the infective larvae of Haemonchus species by susceptible sheep, and also limiting the excessive contamination of pasture land with parasitic eggs that may decreasing the risk of infection. So it is a good approach. Then this approach is confirmed the beneficial effect of rotational grazing against Haemonchosis despite the necessity of differing schedules in accord to the various environments. Actually, in practical condition, in every places it is not feasible to go for this rotational grazing, because when there is properly managed grazing pastures are there, then we can go for a rotational grazing. Otherwise, if the backyard rearing type, or if there is nomadic type of grazing is there in case of animals, then it is very difficult to go for this rotational grazing system. The possible failure to controlling the infection through grazing management can be due to the possible lack of awareness for its potential, as well as for the practical reasons, as animals' movements are determined by nutritional availability. So coming to the diet modification, the ability of the ruminants to resist the parasitic infection when they are kept in good nutritional status has been confirmed. So if the animal is in good nutritional status, then it can resist the parasitic infection. So it has been established that the sheep and goat given a low protein diet seem to be more susceptible to Haemonchus species infection compared to animals given the supplementary protein. The my MBSc work is on giving high protein diet to the goat that has already have clinical Haemonchosis is there. That high protein diet is very much useful in case of resisting this Haemonchus species. So there is a relation between nutritional management and resistance to Haemonchosis, which can be beneficial when it is planned while considering the epidemiology of the helminth, and is applied on its own in a combination with other control strategies. So we have to go for mixture of these strategies, that high protein diet we can give along with the grazing management. It will be more helpful than the one method. Then next is the selection of animals. So we can go for some action of animals with the which having the resistant gene for the this Haemonchus infection. So some breeds of sheep, that is the some exotic breeds that are resistant to Haemonchosis, can be used as an interesting option to minimize the adverse effects of this infection. So that resistant breeds actually have fewer worms than other susceptible breeds. So ultimately they will excrete a smaller number of eggs in the feces, and this will go for less infection in the pastures. So finally, these animals can contribute to controlling the anthelmintic resistance, as they would require less frequent anthelmintic treatments. The introduction of these resistant breeds in our system can be through crossbreeding, which could be a desirable alternative, but should be done with very accuracy of prudence to maintain the production rate of the existing flock. So the possible selection of individuals within a certain breed, that can also be done in our case against the Haemonchus species. Breed within that breed or within our breed, whatever is present in our condition or local breeds, they may be having the resistant characteristics against this Haemonchus species, but we have to go for screening, vigorous screening of the individual animals. Then only we'll come to know about this, whether they have the resistance or not. Because whatever I have seen here, we have actually Assam hill goat here locally in Assam. So they are having some resistant property. I don't know whether it is due to their genetic characteristics or their feeding conditions, or how they graze. If that is the whatever we are doing with the animal, that is the natural grazing one. So I don't know, but we have to go for that higher studies for this, how to maintain the infection. So these genetic selection strategies can be facilitated by the using traits with relatively high heritability. That is the fecal egg count, hematocrit value, and the body condition score, which will give us the their genetic characteristics. Immunity against the Haemonchus contortus, expressed as a fecal egg count and packed cell volume, is a heritable trait in sheep, and there are differences among different breeds in Haemonchosis infection. This has already been stated by various scientists. So some studies in merino sheep breed have concluded that selection of low Haemonchosis contorta fecal egg count may also lead to lower animal production. So in case of merino sheep, scientists have told that if the animal is having low FEC, then the animal is also along with that they are having and lower animal production also. So currently there is one genomic test available, that is Wormster Zoetis they are giving, by means of which the resistance variation among individual sheep can be assessed. But it is in a very budding stage as far as I know. Here we are not using also in Indian condition. So we can think of that to go for genomic test. So potentially in the future, a potential technology could be in the development and commercial availability of the sufficient numbers of transgenic animals can be able to resist the infection. So we can go for a transgenic animals if we get the animals with having the resistance to Haemonchosis contorta infection. Then different product administration. So here we can see the different polyphenolic compounds, the tannins are having the antiparasitic properties. So many plants contain this type of compounds which can reduce the parasitic burdens and increase the animal production. So this can act directly against the helminth at various stages of their life cycle. They improve the immune response of the animals, reducing the shedding rate of the eggs. There is limitations also, as they can lead to detrimental effects when there is they have not been cope up with adequate protein intake. So if there is low protein intake, or if the animal is nutritional imbalance is there, then there will be detrimental effect of this using tannin and other polyphenolic compounds. So one more thing that is nematophagous fungi that is a promising alternative for use against these resistant Haemonchosis strains. So this fungi has the ability to trap and destroy the infective larvae of the helminth after formation of the hyphae. The fungal spores fed to the animal develop during their passage through the ZIT of sheep and predate the infective larvae. So most important species of the fungal, that is the Dodingtonia flagrans, which showed results supporting the use in sheep and goat for control of Haemonchosis contorta. So all these approaches have, when we are using in mixed condition, then only we'll get the result matching with the anthelmintic drugs, because anthelmintic drugs, they are having high efficacy towards these parasites, but due to the resistance only now the problem is arising, and then we can go for using 2-3 control measures to to take the steps against the Haemonchosis contorta infection. Then coming to the vaccination part, as in case of infectious diseases, we go for vaccine production, or we are vaccinating the animals against the disease. So it is a sustainable and efficient option to control the parasitosis also. But in case of parasitic infection, the developing safe and efficient vaccine against these multicellular parasitic worms, it's require a deep understanding of the biology of both parasite and the host, and the biochemical properties of the parasite molecules, and it needs the appropriate tool to assess the host immune responses in efficacy studies, which makes the task a rather challenging one, because we have to know the parameters of both the parasites and the host. So as the biological nature of this parasite is having a large part of the population not being in touch with the host immune system, and with a high reproductive capacity, the major goal of developing anti-Haemonchosis vaccine is to decrease the worm burden in host animal rather than eradicating the parasite. We can go for decreasing the worm burden only. So hence the vaccination of both sheep and goats should not only prevent severe disease conditions of Haemonchosis contorta, but also reduce the transmission via contaminated pastures, which occurs frequently because of the high amount of eggs shed by the infected individuals. So the vaccine that is effective against Haemonchosis contorta has been produced for use in the sheep using the latent H11 antigen, which is a protein of the ZIT of Haemonchosis contorta, and H-gull GP, which are extracted from the adult Haemonchus contortus. This is in commercial use since 2014, but still not. It is not in our market or in our Indian condition. Not much popular here. Now I'm going to give you a glimpse of a case study which I have encountered. Actually, in my MDSC part, I have the animals having the natural nematodiasis, and I have reared the animal in grazing condition only to see whether there is a reinfestation or is there or not, while improving the feed. Means high quality feed has been given to the animal, and then animal is daily they are on the grazing pressures to whether they can check the infection or not. And then I have seen that yes, if we can give the good amount of feed or high protein feed is been given to the animals, they can resist the infection. While they have been mixed with the control animals, all they have been mixed with the infected animals, but still are having the less number of eggs spreading or less number of parasites in the body. So one case study I am presenting here today, it has been in the August-September 2020. Here one occurrence of Haemonchosis is there in our livestock farm complex in goat shed in LCVSCAU Zohing. The flock size is having 22 crossbred and 11 Assam hill goat is there. So here we have seen the clinical symptoms of weakness and diarrhea, and then anemia, anorexia, and when we have observed for the mucous membrane, that is the pale, and we have estimated for hemoglobin percent, it came at 5.4%. So these are the photographs of that case, whatever we have seen. We have done the FAMASA test also to check what is the condition of the anemia. So the animal is highly anemic. You can see in the first photograph that it is a totally white, peppery white mucous membrane is there in the goat. And while going for PM examination, we have seen when the animal are dead, that time after PM examination, we have seen the all over the body there is edematous conditions, when watery conditions are there. Animal is fully emaciated. There is not means only the skin and the very little muscles there. It's very emaciated conditions. We have seen in the second photograph, you can see the hydropericardium. That is water accumulation in the heart itself. And then in third paragraph, it is the abomasum, where we can see the petechial hemorrhage in the third and fourth photograph. There is a lot of hemorrhage in the inside the ZIT. So in this case, the mortality is 9 numbers, that is 8 crossbred and 1 Assam hill goat. Then the morbidity rate is 22 numbers, that is 66.67%. So PM lesions, we have seen the emaciation, anemia, peppery white mucous membrane, large number of Haemonchus contortus worms in the abomasum. There is ascites, there is hydrothorax, hydropericardium, pale visceral organ, hyperemia of abomasal fold, multifocal and ulcerative hemorrhages on the abomasal mucosa with swimming adult parasites. Many parasites are there in the inside the abomasum. So here we can see. So I think you can see the many parasites is there. They are freely moving in the abomasum. It's a heavy infection of this Haemonchus, and we have done the microscopic examination of those these larvae also. It has been confirmed that it is of Haemonchus only. Second, this is also a second video. It is clearly actually visible the parasites. In this video, one more is there. You can see the different petechial hemorrhages this is not from my case, but still we have also seen this type of lot many hemorrhages are there because they are attaching to the wall of this one, and then they will suck the blood. So while attachment, they will do this means all the biting lesions, which is leading to these hemorrhages and all. So it's a very clear indications. In field condition, we used to get this type of lesions only. So in our case, actually, what happens, the animal used to go every day for grazing, and that is also not a managed pasture. From before itself, they are showing the lack of means that in anorexia, and then the feed intake was not proper, then diarrhea was there. So first of all, we have think for the any toxicity symptoms or any other symptoms like that, we thought. Then after that, when we have checked with the pharmacy test, that we got that okay, there's anemia. And then because normally in Haemonchosis there is no diarrhea don't used to be there. So because of the diarrhea only, we are first we are misleading that it may be of some other conditions. But when we got that there is anemia, that is a typical peppery white anemia. Then when the animal died, that time we have done the PM, and then whole case came into our whole picture was clear that it is of Haemonchosis only, because every animal that is having the same conditions, same lesions, etc. So we have given the treatment, and that is of ivermectin 1 ml per 50 kg body weight subcut, and albendazole 5 mg per kg body weight orally, along with a liver tonic as a supportive. And the second step, we have done Zerograzing, that is it means totally a stall fed system we have started. And then we have provided excess concentrate amount we have increased. And then green tree leaves grasses, good quality green tree leaf grasses we have provided in the shed itself after little amount of wilting. Okay, we have not given raw one, just 2 to 3 hours of sun drying, and then we have given the tree leaves so that there should not be any digestive upset also. And prevention in open grazing is the I think main part of this control strategy. So in conclusion, that I will tell that Haemonchosis remain a major problem in the goat and sheep industry across the world. The organism has adapted to survive under diverse climatic conditions. Then resistance has been reported against latest available antibiotics that already field veterinarians, I think they know better than me. Then alternative compounds have also limited efficacy, and their effect on production parameters are least known. Biological control measures have constraints also. So due to these diverse antigenic strains of the parasite, the recombinant production of vaccine is yet a challenge for the researchers. So under these circumstances, targeted selective treatment along with the culling of more susceptible animals and maintenance of good body condition score through the provision of nutritionally balanced diet is considered the best control strategy at the herd level. So the scientists engaged in its related field should carry out extensive research for development of potent vaccine against all strains of Haemonchus controtus, and find a therapeutic agent which should remain effective for decades without having residual effect. So these are the references from where I have collected the different informations. So at last, should we worry for this disease? Actually, no, we should not worry. We should not be over worrying, but we should be concerned about the disease, and we should take the remedial steps at proper time, and we should maintain the proper control strategies every time. If for a profitable farming of sheep and goat, we have to maintain the proper managemental conditions, whereby we can go for zero-grazing method, which is being promoted by Sathyazerograzing, because it is the effective method of to control this reinfestation of parasite in field conditions. Thank you.